and it's the one and only. It's maybe a better phrasing, but so it's extra special for Professor Nel. And he is from André Nel. And he's able to speak Dutch language quite well. We just discovered South African, yeah, Afrikaans, but he is able to pronounce my name very well, and that doesn't happen often with Americans. So André, thank you, and he's director of the Center of Environmental Implications of Nanotechnology in Los Angeles, and we look forward to your talk.
Thank you. Dank je, Gert.
No, you stop, yeah?
And vielen Dank for the Unlading. I'm going to talk today about our system that we've set up called Predictive Tox, and I'll take you through a little bit of the discovery that we've done on nanomaterial safety. So without any further ado, so my talk outline is to tell you, do nanomaterials introduce new hazards? What is the contribution of physical chemical properties other than size? Are there differences in the safety consideration of therapeutic versus industrial nanomaterials? And then how does the high content discovery that I will describe for you in answering some of these questions have helped us also generate new opportunities for nanomedicine. So in brief, how does the area of Nano EHS relate to the world of nanomedicine? They have considerable overlap in the sense that the science is science of materials, but they're very different in terms of regulatory considerations, philosophies, and mandates. EPA regards nanomaterials as new chemical substances which require pre-manufacturing notices. And under the new TSCA reform, that entails mandatory testing for affirmative product safety and the need of data generation for marketplace access and international trade. In contradistinction, FDA does not regulate nanotechnology independently of its existing statutory authorities for drugs, therapeutics, and devices, but does take into consideration properties and phenomena that are attributable to submicron dimensions. So let me first begin then and say something that we have, the discoveries that we've made about nanomaterial properties. And the question is, can we identify unique nanoscale properties and functions contributing in a unique way to biological injury other than small size? And in order to do nanomaterial characterizations, you do need a list of characterizations which are essential to know about. The first is the nanomaterial itself, which, if I can get my pointer correct, is the intrinsic properties of materials, and there are a number of them. The chemical composition, the purity, the shape, the size are all important properties that you need to know and need to characterize. The next is if the material come into contact with a biological medium or an environment, the material changes some of those properties, especially as it relates to charge, corona, dissolution. All of these are important, but in terms of the high throughput discovery that we've launched, I can tell you that indeed there are emergent properties when a nanomaterial meets a biological environment where new material properties emerge that you otherwise couldn't know about. And those are particularly telling in terms of also safety screening and new opportunities for making new materials. So a few of these include the high aspect ratio of materials that can damage lysosomes, band gap in semiconductor nanomaterials. If you, I'll show you in a moment that biology also has a band gap, and the two band gaps can talk to each other. Siloxane rings and silanols in fumed silica as a representative example of surface functionalities that are unique, that come into play at nanomaterials, including surface hydroxides, cationic charge, surface defects, carbonyl radicals, rare earth oxides that combine with phosphates, etc. And then also unique ways of immune perturbation taking place. So taking from what Patrick had then said yesterday at the introductory talk, and I just get myself, is the structure and function of life at the nanoscale is indeed what we were challenged with when we started the University of California Center for the Environmental Implications of Nanomaterials approximately 10 years ago. This was the largest funded federal project on nanosafety in the US, and now after 10 years, I can tell you that it's been a journey of incredible discovery in which we have analyzed more than 400 different nanomaterials. And I can tell you that in this world of discovery, the tools to set this up was done from the inception as a high throughput exercise, because it was very clear that in the emerging of the large number of new materials, you're going to need this. So we had a world-class high throughput screening facility. We generated materials of libraries which consisted of different material compositions. And then we deliberately acquired or synthesized or derivatized them to also give you combinatorial properties where we vary the size, shape, aspect ratio of materials, crystal structure, band gap, surface charge, etc. And the exercise began by saying, well, there are so many different possibilities. We will begin it in terms of cellular functions for organelles, but over time it became smarter and smarter to capture large pieces of biology. And the readouts were all done for computational analysis and then the ability to make a large number of decisions from this. So what had emerged then was the so-called pathways of toxicity. So after finding out what it does to different organelles and different parts of the cell, we could then assemble ways in which it would perturb a cellular function, for instance, generating of oxidative stress, which is a long aspect ratio material, triggering of a lysosome in a macrophage that led to toxicological responses in the lung, for dissolving metal ions, the generation of metal ion species which are released intracellular and generating oxidative stress, for materials that had a band gap, the ability to interact with a biological equivalent and generate oxidative stress functions, for materials that had surface reactivity, the ability to catalyze cell membrane injury with lytic outcomes. Altogether, we built approximately 15 to 20 of these pathways of toxicity, each of which we could ultimately relate for a material or a group of materials to predicting an outcome at an organ level, where the pathway of toxicity became the basis for the pathophysiology of disease. And in the rapid emergence then of this concept, that actually started from nanomaterials, we've now seen a large-scale joining in the concept of adverse outcome pathways. Now, what is an adverse outcome pathway? That is now an intellectual construct that has emerged in the world of chemical toxicity, where materials have a molecular initiating event, which differs for chemicals and nanomaterials, and then a series of key cellular events which could involve gene activation, protein production, that are linked to an adverse outcome. And these different levels of key events and adverse outcome describes then a series of events at molecular, organelle, organ outcome, then individuals, and in the case of the environment, also population outcomes. So that the adverse outcome pathway, instead of having to go through all the steps of prediction, now allows you to take information that already exists in the literature and in the toxicology database to construct AOPs for decision making. So let me give you an example here in Europe. Best described is the adverse outcome pathway for skin sensitizing chemicals. You're no longer allowed to do animal studies. Out of the actives, dyes, flagrances, it's very important to say which of these materials comprise materials that can lead to skin sensitization. And that's premised on electrophilic chemicals, which under catalyze covalent binding to skin proteins. This is the molecular initiating event, for which there's a simple test that can be done, a direct peptide reactivity assay. Once the keratinocyte is compromised by the covalent binding, it will send out, it will generate oxidative stress, and it will send signals which bring in dendritic cells. Both the keratinocyte can be assessed by an oxidative stress assay, a straightforward, simple assay. The dendritic cell can be interrogated by an antigen-presenting cell activation assay. And in terms of the entire construct, this means that the education of a T cell in the local lymph node will ultimately go back to the skin, where it will cause skin damage. And the way that this now assists decision-making for these chemicals, in one example of the AOPs is by Dr. Landsiedel, who proposed this for BASF, where you would then ask the three questions. You do the direct peptide assay and you will do the keratinocyte assay. If the answer to both of these is yes, you can classify that chemical as a sensitizing substance. If the answer, say, to the first test is no, you will do the third test, and if the answer is yes, it may lead to acceptance. So the debate about exactly how to use this is still ongoing, but the decision tree is real, in that we've now covered a huge amount of ground in being able to do these characterizations. So let me now tell you how it works in the world of nanomaterials. As one of the large number of materials that we've examined, a frequent material encountered for regulatory purposes is carbon nanotubes. They come in lots of different flavors with different properties. And how do you bring order to such complexity? Well, we did this again through the acquisition, synthesis of a large number of nanomaterial libraries, from multi-wall, single-wall carbon nanotubes, long, short, functionalized, non-functionalized, etc., where the screening for a molecular initiating event, you can have various different outcomes, adverse outcomes. For instance, malignancy is one possibility. The one that more often triggers concern is that workers could be exposed and that you could get lung injury. So from the perspective then of, say, lung damage, a key central molecular initiating event is the ability of these materials to generate lysosomal damage. And from this construct that the material has to get into the lysosome, there's a series of property-activity relationships that belong to these materials that will determine that outcome. Once it's localized in the lysosome, surface catalytic activity, surface functionalities, oxidative stress, metal impurities will then determine whether you damage the lysosome. So what happens if you do damage the lysosome? Well, the adverse outcome pathway is then dependent on the triggering of this event at a macrophage in the lung, where the production of the lysosome damage lead to assembly of an inflammasome, which produces interleukin-1 beta. And downstream of this event, once this is triggered, subsequent responses in the lining bronchial epithelial cells lead to a process known as endothelial mesenchymal transition. Basically, the epithelial cells transition to collagen-producing fibroblasts with the production of TGF-beta and PDGF. And so if you screen in terms of an in vitro system for the production of interleukin-beta, TGF-beta, PDGF, there is a close correlation and a predictive outcome for any carbon nanotube that you will test in terms of whether it will complete the cycle and lead to collagen deposition. That means the outcome of lung fibrosis and collagen deposition is directly equivalent to what you can test in cells, and you can also discern these responses in the lung at various points in time. So, give you one example. One of our libraries is multi-wall carbon nanotube. It came in a crude form. We did surface functionalization, carboxylation, pegylation, added NH2 groups, or added a polymer with a cationic surface. At the level of testing for the lysosome damage, you can see here, that's the crude tube. The intact liposome is a little dot. So material number 1 triggers the lysosome, produces interleukin-1 beta, and in the animal lung, there's clear evidence quantitatively of collagen deposition and lung fibrosis. Contrast, materials 2 and 3, lysosome stays intact, very little interleukin production, very little lung fibrosis. If you now have a slightly cationic tube, more lysosome damage, again, you get a correlation, a predictive response at cellular level and then lung outcome. And if you progress to a material that is highly cationic, such as the polymer-coated particle, you can clearly see that reflected as well in the cellular outcome. So out of this, you can now construct a system of an adverse outcome where the original screening test for the NALP3 inflammasome activation could constitute your molecular initiating events. You have a whole series of structure-activity relationships that you can relate to that, and with time can lead to the classification of materials at that level. At the level of intermediary testing, you can do the screening for interleukin-1 beta, TGF beta. So this could be for one key event, the TGF beta and PGGF could be for another key event. In the era where we still live, of animal testing, you can do the short-term animal testing to confirm these outcomes. And what is currently happening at the world of regulation, you may be asked for a 90-day inhalation study, which is going to be considerably more expensive. You will be perhaps be able to test one material, cost of $500,000 to $1 million, whereas these other tests, this can be performed for a few dollars and this can be performed for a few $100s. So this is also called tiered decision making. And in consideration of how this can be used, the knowledge, this was actually co-published with people from industry, people from EPA participated, including the head of the OECD in the US, where you could then say, all right, now you can make a decision tree, which can begin by asking the important question, is this material of concern to humans? And in the case of respiratory toxicology, yes, if it's inhalable. Is there a potential for exposure? If the answer is yes, then you need data. And now you can ask the data in the current context, if you can do a tier 1 test. And if that's not enough, you can demonstrate the fibrosis in the lung, or you can go to the tier 3 test. In the way that it is intended to be used in the future and can lead to a considerable facilitation is that you use the key event testing as I've outlined you. Test 1, test 2, and a test 3. And so we've done this type of exercise for more than 400 different nanomaterials, and the predictive science and the intellectual construct can be used for many different nanomaterials. I show you here a category of metal oxides, where you can go from the soluble metal oxides, transition metal oxides, rare earth oxides, long aspect ratio, materials with surface reactivity. There's more than 90 different materials that I show you here, in which you can do key events, outcomes that are predictive, and which we tested in the full prediction scale before we started working on writing it into AOPs. And then as a result, you have a whole series of key events, and you have very specific molecular initiating events that you can choose And that will give you a test immediately to perform to instruct you what the possible decision is that could be made about the safety of this material. So I'm not going to go into the complexity of this, but in terms of just the metal oxides, there's a whole construct now in terms of chemical toxicity where these relationships between different triggers and intracellular organelles can be reworked into key events with databases from the literature in which you don't have to do the full prediction, and that can assist then in the decision-making process. So what is the reality? Well, as we move with the TSCA reform, which took place last year, there's now, for the first time, mandated that safety evaluation of all chemicals take place in the US, that all chemicals be evaluated on a science-based standard, and that affirmative chemical, physical, chemical, and toxicological data collection needs to take place before a material can make it to the marketplace. The language includes strong indication then for the use of the non-animal based alternative test strategies that I've outlined now, in which the language call out for reliable alternative tests on animals, high throughput screening methods, predictive models, computational toxicology, and tier testing. And we were fortunate, Tim Malloy and myself, is we read the bill, we commented on the bill, and Tim even wrote write-ups in the bill. So what are you going to do with it? Well, if you take the world of decision-making regarding material safety, there's always the complaint, but we don't know what to test. We do not know what data to generate. And that leads to a continuous discussion of how do we go forward. I think that we have a potential construct that could be useful. The question is now, do we have the skills and implementation to make it work? Because while this exercise was carrying on for engineered nanomaterials, the rapid evolution of the science from nanomaterials through nano intermediates and then to nano-enabled products didn't stop at having an industry for production of nanomaterials. What was nanoparticles quickly became absorbed into materials that is used for energy, for environment, and so quickly lithium-ion batteries and their content became the products that we now currently need to be concerned about. And adding to this is that under the new EPA rules, EPA have now also brought out a rule that materials that is made deliberately through size, coating, shape, surface area, etc., to perform their function are materials that could now be asked to generate data. And how are you going to generate those data if you don't have a simple construct? A whole new group of actors, grinders, mixers, formulators, dispersing scientists are all going to be called in. And this already is causing great concern in the community of commercial products. So it's almost as if we were running and missed the train, and now the train is somewhere else. It's a virtual train in the sky, and we have to join it. In my opinion, this is one of the ways in which we can overcome that deficit. And why is it important? Well, I'll show you one example of why it is so important. When carbon nanotubes began, it was touted as the next wonder product, $1 trillion industry that could emerge. Unfortunately, it never happened for a lot of different reasons. Hype, overproduction, the materials were not pure enough, but one important part was when carbon nanotubes were described as fibers, which was not entirely accurate. It was based on a specific material. There was a carbon fiber and not a carbon nanotube. It caused a lot of the industries to drop their manufacturing. We're now at a stage where here is the carbon nanotubes where we have an equally important material, graphene, that is being produced for many different technologies and also stands to become a multi-billion-dollar and even a trillion-dollar industry, depending on whose predictions you follow. And so I can just say, taking the exercise then of what I've described to you, we've already made our libraries of graphene in various different forms and can perform predictions about its interactions and have developed AOPs that could be implicated. Do we have the will to do it? Well, time will tell. So if you now move to the world of nanotherapeutics, the science of physical chemical characterization, still very important. In vitro testing, although a lot of the in vitro testing also looks at pharmacology, drug aspects, the big difference from Nano EHS is here you have to do mandatory animal testing. So you don't have to skip this step, and also the volume of production of materials are lower to allow the animal testing indeed to be an important component. And then in screening, you could use many of the same screening principles that I've shown you, but therapeutic nanomaterials do introduce hazards not seen commonly and screened for in non-therapeutic and commercial nanomaterials. And they listed here things like the complement activation, anaphylactoid reactions, effects on blood coagulation, platelet aggregation, hemolysis, cytokine production, sterility, etc. Scott McNeil have outlined the wonderful NCL assay cascade that they've developed to take you through all these different steps. It's done deliberately. There's a comprehensive list of tests, but they do it in a stepwise fashion in order to inform about nanomaterial safety very appropriately. So what I'm going to tell you now in the last few minutes is, so what is the benefit to nanomedicine of all the things that I've told you now? Well, we're continuing to learn. For instance, from liposomes, the quantity control methods for the liposome lipid composition, the state of the encapsulated drug, the density and conformation on PEG, these are all items that, in spite of a material that was already approved decades ago, continuously leads to new methods for development to understand this, because this is important in terms of outcome. And we also become aware that while it has been a very successful and still growing industry, there are shortcomings of liposomes. For instance, that it has an unsupported lipid bilayer, which can lead to leakage. And in terms of leakage, if you take the innovation recently made by Onovid, which is an irinotecan delivering liposome made for protected encapsulation to improve delivery and to reduce toxicity, did get market approval, but a black box warning for safety. We argued that a lot of the aspects on the safety, which it could not totally correct to improve entirely over the free drug, was due to the leakage, and used our knowledge then of building material libraries to develop a liposome equivalent, which we call a silicasome. It's a mesoporous silica nanoparticle coated with a lipid bilayer. It has a huge drug loading capacity. It's not just passive entrapment now of a volume, but you can actually bind the drug to the pores of the walls through a variety of different chemical interactions. You have a much higher loading capacity, you have less leakiness, and you have more stability. And the outcome in testing the silicasome loaded with irinotecan against the liposome was that not only did we get a more efficacious drug, as you see here, this is a rigorous animal pancreatic cancer model where you see the tumor and the metastases, free irinotecan. Here's the liposome and the silicasome. You can see there's a considerable improvement in the outcome, including even a survival outcome for some of these animals. In terms of that safety issue, the free irinotecan and the liposomal variant induces liver necrosis, induces apoptosis of the small intestinal villi, and that's a well-recognized serious side effect of the drug. It leads to an acellular bone marrow. The Silicasomme had none of these problems. We only had a mild amount of steatosis, no gastrointestinal abnormality, and no bone marrow toxicity. And if you looked at the studies of the modeling that we did on this, the explanation is that the liposome has after being captured by the Kupffer cells, for instance, have a much higher rate of degradation, where the leakage of the chemotherapeutic into the hepatocyte overwhelms the metabolism of the drug, whereas in the case of the Silicasome, the slower release can keep up. We were also then, in the context of making the libraries, had to prove the safety of the new form of silica, and we acquired a library of materials that involved making all the forms of silica, and then being able to figure out that in the end, what can make silica toxic is if it's made under high temperature, where you have chain-linked particles that binds to the cell membrane, which have a highly energetic siloxane ring that opens, that these are the constructs that lead to membrane damage, which also triggers activation of the NALP3 inflammasome and can then set up a whole series of toxic side effects, which we carefully screened by predictive tox making. And mesoporous silica had none of these outcomes or none of these effects. A third major difference between mesoporous silica and the fumed silica was also that fumed and Stober silica undergoes a bioretentive. They often remain for long periods of time, whereas the mesoporous silica breaks down into silicic acid within 7 days, and metabolically you can recover 98% secretion into the urine. So to end up my talk, I see my time is up, is that out of the high throughput discovery that I've shown you now, a host of other very useful products came to the foreground. For instance, we've learned from the world of nanomaterials there are several ways in which you can induce cell death, that the induction of some of these libraries of materials or delivery devices can induce immunogenic cell death. And a hot topic at the moment is cancer treatment with immune intervention. And I don't have time, perhaps I can show you this next year, an astonishing outcome, jaw-dropping, in making use of these properties of nanomaterials for cancer treatment, in addition to the innovation with the chemotherapeutics. From the side of, say, adjuvant materials, alum being the gold standard for all the years. It's an amorphous particle. It works via the NALP3 inflammasome. Out of the screening comes, if you make aluminum oxyhydroxide, which you make in the forms of rods, polyhedrons, or plates, that they have quantitatively the ability to drive the inflammasome harder and make an order to 2 orders of magnitude better immune adjuvants. I can take you through our discovery process of taking nanoparticles to places in the liver where they can encounter different aspects of the immunoregulatory cascade with a view to developing new treatments for allergies and autoimmune disease. I can take you to the discovery that I showed you about graphene oxide being able to be functionalized and insert itself into membranes in various different formats, to developing a graphene oxide library that quantitatively destroys bacteria by cutting them up into smaller pieces which you can put onto catheters. So, to give you my last slide, what then started as an exercise of nanomaterial safety screening with some of the best material scientists in the country as part of this group, engineers, biologists, computational toxicologists, has then led not only to a large-scale impact on nanosafety, but what we are also being able to do is to take the knowledge that comes from that and develop multidisciplinary team science, such as the efforts for the multidisciplinary pancreas cancer treatment group that we have. Thank you very much. I'm happy to take questions.
Thank you very much for leading us through an impressive amount of data and also confronting us with the problems of decision making, which is always not easy. Even the more you know, the more problematic the decisions probably are, and also showing us how to go on. And I would like to ask you, please react and comment.
Congratulations to this great work and great talk. Mm-hmm. I have a question about the relationship between your testing strategies, which try to avoid unnecessary testing if you can identify a pathway, versus the testing cascade proposed by NCL. This seems to be a fundamental conceptual difference. Do you see a way to kind of...
No, I don't think we're going to easily change the mode of thinking in terms of making a new drug in which the tradition is not. They're not looking at the technology. The tradition is you will do whatever testing is necessary. And as part of that, animal testing is absolutely essential. I don't see that going, I'm not seeing that will depart very soon. If you ask me, is the constructs that we've developed being able to pinpoint from a material key physical chemical characteristics for which we can do rational testing in which you link observations at molecular level to what may happen in a cell and then what may happen in an animal, I think there's a good chance that that will happen. If you talk about the world of Nano EHS, commercial nanomaterials, in that particular respect, there is the dire need to get away from animals simply because of the large number of new materials being produced, the cost of that testing, and the philosophy of how it was done in the past, descriptive animal studies instead of mechanistic studies. Is that going to happen immediately? No. I think these AOPs, which are now driven by OECD, is a step in the right way. In terms of the new mandates for the TSCA reform that has just taken place, remember we have 80,000 industrial chemicals, fewer than 1,000, that have ever undergone testing in the past. So you can imagine the necessity of finding ways of doing it without reverting to animal testing. But it's an active debate. It will take some time. With the new guidelines coming from OECD, the AOP wiki which have been established, and the actual concept that you can have a construct of data generation which is linked to an outcome, in my viewpoint, is the correct way to go with this.
But this argument says essentially that we do animal experiments because we always did animal experiments and that will not be changed soon. On the other side, I think the CLINAM event is a platform where we are happy to have FDA among our ranks to discuss exactly these questions. The bad side about all these animal experiments is that their relationship to human pathology is somewhat limited. We see it everywhere. And so it's painful to do animal experiments which are ethically not questionable, but the question is, are they always necessary? And at the same time, they are time-consuming, they are money-consuming, and they contribute significantly to the very high price of new drug development. So maybe I can ask the question to Frank Weichholz sitting here, what he thinks about the potential of a paradigm shift in assuming such strategies in the future?
Sure, if I'm not a little overwhelmed by this complex question, because it's also a question of validating the systems in context of use. So since the materials are quite different and the challenges can be very different, of course we like to have a battery of established principles, as you illustrated. Some of them which are derived from modeling exercises and knowledge that is obtained from databases, and we get better and better at that. However, I think there has to be a validation step in between, and I think we need to be mindful. So I'm not sure if I can answer the question.
I think that that's absolutely correct as I see it from the world of Nano EHS, commercial nanomaterials. Validation is still a very big hurdle to overcome. Even after validation has taken, has been established and have been accepted, it will take a considerable amount of time. But the sheer volume of these materials as they're being introduced, I think is more or less going to force the outcome of making decisions with simpler tests and then go on a tiered approach. Once you've made the decision, you can then always decide that I'm going to have an additional tier of testing in order to go further. For FDA animal testing, I think it has proven its value for drug development, that you do need animal testing. And we all know the statistics that even with the best animal tests, you still come up with surprises after a drug is introduced into humans. I don't think we're going to go to eliminating animal testing in terms of drug development very soon.
Thank you so much. I got orders to stop this discussion. It's a very important discussion, and it will be continued, not only in future CLINAMs, but I think in many meetings. So thank you very much. I highly appreciated your talk. I had some burning questions myself, but we have to do that during dinner or so. Yeah? Okay, I have to make place for the next chairman. So I cannot introduce you, Avi.